Hello, everyone. As I said, my name is Danu Tsirimia. Thank you, Christoph, for the introduction. So today I will go step by step in presenting you how to do the offline analysis for recoveriX datasets. Okay, so on my screen, I haven't created slides, but I will do an online and step-by-step demonstration. So the first step in here on the screen, we see the g.BSanalyze software. And the first step would be to load the recoveriX dataset. So I will load, for instance, this file. And now the raw dataset recorded with recoveriX will be loaded and displayed into g.BSanalyze. Okay. So when first loading it, yes, we have the raw data set. We do see a lot of markers and so on, but the EEG is not very clear because in this case, the data is not filtered yet. And there is a lot of DCs, so the screen cannot, the screen is not auto-scaled right now. The first step in processing this data is to filter it. g.BSanalyze provides a lot of filtering options. So by going to filter, we have the opportunity to design different kind of filters. So for recoveriX, we will need to design a filter in a bandpass filter, Butterworth, in between 8 to 30 Hertz. So of course, BSanalyze allows us to develop different kind of filters. So low pass, high pass, band pass, and band stop. And as I said, I will select the band pass filter, and the filter realization type, it's in our case Butterworth, but yeah, we have many other options. The lower cutoff frequency of the filter will be 8, and the upper cutoff frequency will be 30. And I will set the filter order to 5. And I will just rename it filter recoveriX. I will add it to the list. And okay. And we'll exclude the last channel because it's the validity channel. And I will exclude it from the filtering and we'll click on stop. Now we can see that the data is filtered. It's a bit nicer, but I will go forward because we want to see every single trial. So we will split the raw data into trials for left and right imaginations. So I will go to the trigger window where I will select the trial parameters, which are the time before the trigger. In our case is 2,500 milliseconds. After the trigger, we have 5,500 milliseconds. And I showed you earlier that we do have different markers for left and right trials. These are in the data set. So I will trigger this data set based on the trials, based on those markers. So I took the right marker and I will name the trial as right. And we'll add it to the list. Then the left one, rename it left, and add it to the list. Okay, and I will start the triggering, and now the dataset is triggered, and we can clearly see now the very first trial, which is the right one, and we can surf through it. So we can see that all the channels look very well. I will set also all 16 channels to be seen on the screen. Okay, so having a very fast look through the data, we can see that it's very nice and clear. Okay, I will go back to the first trial. A very important part for calculations for recoveriX and the CSP, basically, for motor imagery, it's the electrode disposal on the scalp, because we also have during the processing, we also have to perform different source derivations and so on. We have montages for every single product that we have. So in this case, we'll use the recoveriX montage that I will show you right now. So montage creator. So right now I will load the montage that we used for recording this dataset for recoveriX. So now the montage is loaded. We can see that the C3 and C4 have different colors because these are the main electrodes, because C3 overlies the right hand motor cortex projection, and the C4 for the left hand. And in here we can see the source derivations for the C3 electrode. We have a secondary electrodes, the FC3, C5, C1, and CP3. And for the CP4, the others. So I will close for the moment this montage. We'll go back to our window and we'll load this montage to the dataset. So geometry, we'll browse again for the same montage files. Now we'll wait a bit and we'll see that for every single electrode, we will have the positions loaded. So now they are loaded and we just hit OK. As I already stated, we have to perform a source derivation or the so-called re-referencing. Okay, so source derivation, and I will replace only the involved channels. So in this case, as I told you, we'll do a Laplacian derivations for both C3 and C4 electrodes. Now, after applying the source derivation, we can see already that channel number 5 and channel number 9, which is C3 and C4, have Laplacian labels just beneath them. A very important thing is to have a look in the spectrum. So we can go to the spectrum and check for both for C5, for C3, and also for C4 to do a classical spectrum analysis. So I will use a 1000 milliseconds window. I will do a comparison in between the reference and the signal, and in here we'll have 500 milliseconds. The reference interval will start at 500 milliseconds, and the action interval will start at 2500. Okay, as a result, the data window is Henning, and we'll do it only for channel number. We'll include it only for the right trials, which is channel number 5C3. So I will include only channel number 5. Okay, and start. Okay, in this window we can see the spectrum. Of course, everything right now it's scaled on logarithmic scale. I will switch it to linear, and of course only up to 40 Hertz, because we don't need more. And Y scaling, linear, scale. Okay, so we see the spectrum of the reference interval with blue line and the spectrum of the action interval with the green line, and we clearly can see the difference in between them. In the same way, we will do it also for the for C4. So all the settings remain the same, and I will just include only the left trials. And in here, channel 9, it's for C4. And OK, and stop. Again, I will switch the scales because for these low frequencies, we don't need logarithmic scale. And linear scale. And here we have it for for the C4 channel, for the left hand. Again, we can see a very big difference in between the two spectrums. Okay, for the moment, I will close these two windows. And I will go further to the ERD, to the event-related desynchronizations. These are, so the event-related desynchronizations, my colleagues already explained you that appear when when a user imagines or does a certain movement, they appear at the projection of that feet on the motor area. So I will go to event-related desynchronization. First, I will set a start reference period at 500 milliseconds and the end of the reference period to 2000. Okay. And I will design a new filter, which is a bandpass filter. Now it will be an FFT filter ranging from 10 Hertz to 12. Okay. And I will rename it. And we'll add it to the list. And okay. And now I will include only the right trials. Include only right trials and channel number 5. Okay, and start. And now in this picture, we can see the event-related desynchronization for the right hand imagination. So we can clearly see that in here, the time course went down. We will do the same also for the other trials, for the other classes. The filter will remain exactly the same. And I will only switch I will include only the left trials and the 9th channel. Okay, and start. In here, we can see it a bit more clear that in the left side, we have the reference part, and on the right side, we have the desynchronization. So in this time, the right vertical line is the trigger when the cue was presented to the user, and he started imagining the movement, and we can see that the event-related desynchronization went down. Again, a very important step from here on is the ERD maps, event-related desynchronization maps. So we can calculate them again from the Analyze menu. We will set again the start reference period at 500 milliseconds, the end at 2000. The lower frequency border will be 8 Hertz because this is how we filter it. The upper one, 30 Hertz. Bandwidth of single bands 2, and step to shift the single band to 2 Hertz. subtract phase locked signals only, and the significance test bootstrap. And let's select it first for channel C3. So I will include only the right trials and channel number 5. Okay, and start. So in this figure, we can see clearly that the desynchronization happened at about 18 Hertz in time from second number 4 till the 8th. So they are very nicely seen in here. Let's do the same thing also for the left hand. So the settings will remain the same, and I will only select the left trials and include channel number 9. Okay. And do the same. Okay, also in here we can see that the desynchronization appear in also in some other frequency bands, okay, from 8 to 10 and so on. And this is a very good point. Okay. And the next step is to calculating the CSP filters or the common spatial patterns. Okay, so I will go to CSP. And first of all, I will select the first class, which is right, and the second class, the class number 2, as left. So the CSP filters have to be created from a certain time window, from the feedback phase. So I will take it from, let's say, from second 5 of the trial, from 5,000 milliseconds to 7,000 in this case. Of course, this window can be shifted. So from 5 to 7, or to 5.5 to 7.5, or from 6 to 8, and so on. And I will save these results. So okay, and start. Just one moment, because I forgot to exclude the channel. I forgot to exclude the last channel, which is the time, the validity channel. And now we can see these heat maps. So with very, very small numbers, we have the electrode disposal on the scalp. And we have these spots in here at about, in here it's the C3, right near C3 electrode. And in here, somewhere about the C4 electrode. Okay, anyways, I will show it to you a bit later with in 3D. Okay, it's saved. And right now I will go for spatially filtering all this data set. So first I will exclude the last channel because we don't need it anymore. Transform cut trials in channels. I will select the last one and start. And now I will go to specially filter this data set. I will select the file that we just created, recoveriX CSP. Okay. So now it is loaded, and I will use the first two and last two spatial filters, which is 1, 2, and 15 and 16. And I will replace all the channels. Okay, after doing this, we have now only the 4 feature channels. So there are publications stating that when using the CSP method, you only need 4 special filters for that. The first 2 and the last 2, which are the most significant filters. From this step on, we will have to compute the variance. So this is done going to parameter extraction, variance, and I will take a segment of 1000 milliseconds, so 1 second, and the overlap should be 996, 1 sample less. So in the upper window, we have the time, the length in milliseconds, and down in samples. So I will take only 249 samples, 996 milliseconds. So I will replace all the channels with the variance feature. Okay, now the variance is calculated, and the next step in this whole processing chain is the normalization. The normalization is done via the arithmetic function, so we have multiple possibilities in here for all the channels: addition, subtract, multiply, z-mean, normalization, and so on. Right now, I will select only the normalization, and we'll click on Start. Yes. Again, another step is the log10 processing. Okay, so I will select the log10 and apply it to all the channels. Start. Okay, now the data set is fully processed up to here. And from now on, we can start creating the future matrix, the classifier, and classifying all this data. So I will create the feature matrix first. The classification interval should start at 500 milliseconds with a step of 500 and end at 8000. I will select the right and left classes and we'll select the feature channels, all these 4. Okay. And the classification method will be LDA, Linear Discriminant Analysis, and I will also save it. And we'll click on Start. Now the classification window popped up. So we can calculate it as 10 by 10 cross-fold validation to see the cross-validated error rates. So I will map the feature number 1 to the 4 and start. So this is the classification window, but I will show you directly the CV error rates. So the cross-validation error rates, the first graph in here, it's the total error rate, which is basically the sum of the classification error rates for class number 1 and for class number 2 for right and left trials. And now I will create the classifier for 100, train 100, test 100, and we'll save it. recoveriX classifier, and we'll start it. Now we do have the same error rates, almost the same, a bit smoothed. Okay, and on the first part, okay, I will close this because we don't need them anymore. We'll create a VLDA. And now I will go next to apply the classifier on this dataset. So classification, apply classifier. And now I will have to load the classifier that I just created. After loading the classifier, it's very important to, sorry. It is very important to select the classifier with the lowest error rate, like this one in here. Okay. So it's not time 6000, and I will click on start. Okay, now we have all these channels, all the data set is classified. So every single channel in here, it's the classification output. And now let's go further for checking the output of the classification to see exactly the error rates. Classification output mapping. So I will select the right and left trials, the right and left, okay, and the first two channels. Okay, and start. So this is the mapped output of the classification. So with dashed line, we see every single trial that is plotted in this graph. And with solid line, with green, we can see the average of those of the trials. So after the trigger line, which is the red line, we can clearly see that they are pretty nice separated from the middle line in here. Okay, same in this graph, we can see that again, for every single channel, they are going down as they should be. And in the third graph in here, we can see the performance. So the accuracy in this case, so for with green and blue, we have the accuracies for the right and left trials, and with black, we have the total accuracy. So at a certain point, we can see that almost at 6.5 seconds, the accuracy goes up to almost 100%. Okay. Regarding this happened regarding the accuracies. And the whole processing chain for the recoveriX files. What I do want to show you more is how to define and how to nicely view the electrodes in 3D. I will load again and I will run and load again the montage creator. So we still have the same montage loaded as previously. And I will show it to you in 3D, and I will explain what's happening next. So sometimes it's pretty difficult to see in 2D how it is and where are all the electrodes located and so on. This is why we went further in creating a 3D tool also for g.BSanalyze that allows us to see every single position of the electrode. So in here in the left side, we have the main view, which is rotatable. And on the right part, we have the three projections. By turning it, we can nicely see every single position. And from now on, I will show you what we could do and what results we can plot on this column. Okay, so I will close this, and I will go back to the CSP part in order to plot the common spatial patterns directly on this file. So I will load the dataset again, recoveriX. So everything will be faster. Now I will go through the whole processing chain that I did a bit faster. Data, it's not here. It loads the file. I forgot to mention that g.BSanalyze has also a batch mode. So all the processing that I did earlier, all the chain, for every single function, it outputted the code in order to allow the user to perform everything via code. So if you have multiple files for processing, you can do it automatically by copying this code into a code file, into a script. Okay, so at this point, I will just go very fast to the filtering in order to, okay, 8:30, start, exclude this one. Okay, triggering. Two 2.5, 5, 1, right, and left, list and start. Okay, we'll load the montage. Geometry. And we'll go to classification, to common spatial patterns. So the first will be the right, the class 2 will be left, and we'll take again the 5,000 till 7,000. And this time, okay, I will show it with both files, with both visualization methods. First of all, with the 2D tool, and second with the 3D tool. Again, I forgot to exclude the last channel. I will do it very fast again. and channels and the last channel. Okay, so again, two common spatial patterns. And 5,000 to 7,000. Show with both result 2D and 3D. Let's start. Okay, now we can see these two very nice spots in here. And the 3D tool, which will display everything on the sculpt. So we can see that the red spot in here, it's beneath electrode CP1, right near to C3 and CP3, right here. And I will just move it a bit. So they are quite near in here. And for the other part of the head, okay, I will rotate it also in this part. So now, right now, it's very clearly beneath C4, as also in here, in the very last CSP filter. Okay, and the trick, the very nice part is that we can also plot it now directly on the 3D brain. And now with this, in this part, we can nicely see it beneath the C4 and directly on the brain. So yeah, the electrodes are located there, have a couple of millimeters distance from the brain. And now, as I told you, you can see very nicely the activation, the CSP beneath the electrode, beneath C4. Of course, everything can be also, we implemented a part for exporting figures. So we could export the main, all the axes in a figure, or only the left one in here, which is rotatable with different resolutions. So for instance, I will take 300 dpi and I will set its name, C4, and save. Okay, and now the picture is saved in the same working directory on the path that I set. And now I will open it outside MATLAB and we can zoom in and zoom out. So to be honest, this is a very nice feature for different journal papers and so on. Okay, so this is a whole process. This was the whole processing chain for the for a recoveriX dataset. Another very nice point for brain-computer interface users is the event-related potentials. So the so-called P300. So I will show you now how to nicely plot and to check the P3s from a dataset. So I will load a P300 dataset, which is already triggered. Oh, okay. So for every single function that we have in g.BSanalyze, we also have data sets for them, and also very nice demonstration, very nice examples. So in this case, we have a auditory evoked potentials. I will show you how the data looks like. I will display, let's say, 10 seconds of data. So you can see that there are targets and non-targets. Okay, and the markers when the trigger occurred. And I will use the average function to display, to show the P300 evoked potential. So I will use a baseline correction in between 1 to 26 samples. And I will do it for target and non-target and use a statistical analysis. And now we can see the resulting window. So up here we have, with green line, we have the P3, the evoked potential. The red vertical line is the trigger when the event was displayed on the screen. And from here we can count. So we can see with this blue patch, in this case, the difference in between the target trials and non-target trials, it's statistically significant. By target trials, I mean when the user focused on that stimulus, and non-target trial is when he did not, when some other distractor was played, or when, if we are discussing about a P300 speller, when he watched, I don't know, when the other characters flashed. Okay. So in this case, we can go measuring all this. I will take the second graph. I'll go to measure. And in here, we can click on everything. So we have the N100, and I will click also on the P300. So it's almost on the peak. Yeah. Okay, so the P300 stands for attention. I think my colleagues already told you about all this. In here, it's very nice that we can measure exactly the latency. So this is the peak up here. So we can see on X, we have 343 milliseconds. Of course, this can vary from one person to another. So let's say that if the person is a bit older, the P3 latency is higher. So it can go up to 350, 360, and so on. Again, also the amplitude is a very important factor in P300 in evoked potentials. So in this case, at the second marker, we have the amplitude like is 7.71 microvolts. So we can measure all this, and yes. So I will go also at the P3 channel. No, I will go to FPZ because on the first two channels we have the highest amplitudes. Okay, so the third one, the latency, it's again 336 microseconds for the second one. Yeah, probably it's how I've clicked on it. Yeah, it's again clicking on the graph. So this is the very nice tool. Again, we can see in here that the small patch it's green and the part this part it's blue. So the green means that it's statistically significant. So the p-value it's below 0.05. And for the blue ones, it's highly statistically significant, and the p-value it's below 0.01. With the gray part in here, it's similar, only that it's negative. So the difference is negative in between targets and non-targets. So the p-value for the light green part, it's below 0.05, and here for the gray one, below 0.01. So up to now, I showed you only a very small part of the features that g.BSanalyze can calculate, can do, and so on. There are different parameters that we can extract from a data set. We can perform like Short, Barlow, AR, variance, bound power, exponential window, and so on, cross correlation. And I'm waiting, so I will give you more details for this when in the Q&A session. So I think I was pretty fast with my presentation. So if there are questions, I'm happy to start answering to them. Okay, in chat. Going back to the trials part, so I showed you earlier exactly how to split a dataset into different parts. But for instance, if there is a single trial that contains artifacts, we can mark them all. So artifact, we can select the marker from here and mark this specific trial as an artifact. Okay, I will display less trials, like 4. Okay, and going through the data set, we said that, okay, this one looks like an artifact. I've marked it as an artifact, and it poops up in here. And I will mark another part. I don't know, let's say this one here. I will click it as an artifact. So if we do want to clean the data set, we can go cut trials and channels and remove everything. So I will select only the artifact, exclude artifact, and okay, and start. So right now, all the trials that were marked as artifacts were excluded from the dataset. Of course, the triggering part has also some other options. When we could also line up some trials if we have a raw data set that we want to process. Let's say we don't have markers in it, we can split it into different epochs. So I will import again the same data set. It loads. Okay, so considering that we don't have this these markers in here, usually at the very beginning of the file we have some amplifier drifts. So we can take out the very first part of the data set, like let's say like the first 10 seconds of it. So I'll cut data before 10,000 milliseconds. Yes, I want to remove it. And now it's clean. So we can go to trigger again, and we can use the lineup trials. parts. So let's say that we do want to split this data set into equal parts, let's say of 2 seconds or 500 samples. So as I said, line up trials and start. Right now, the whole data set is splitted only in equal parts. So let's say 10 seconds. Now we have 5 trials on the screen and so on. And we can do further processing and exclude different parts and so on. Again, we have the epoching part. So for instance, if I'm interested only in a certain part of the data set, let's say only this epoch here, I'm selecting it. Pretty difficult to see. Okay, now it's done. And I will go to analyze. So for some specific artifacts or data sets, we can check directly the spectrum in here. So we can see it. So it goes up to 120 Hertz, but I will select some more. Okay. This one, also this one. Okay, and analyze. Okay, so we can see very fast the spectrum in here, or even the raw signal. Okay, so thank you very much for your attention up to now. Here comes the questions, the Q&A session. Okay. Until I will get some other questions, I will load another data set where I can nicely show you a bit better this analysis part. Okay. So I will select this epoch here. So this is 1, 2, and 3. And the fourth one. And we'll go to analyze. Okay. Now in this case, we can nicely see the spectrum. So at about 3 Hertz, and here it's something like 17 Hertz. Okay.
Danu, there's a question, which was, could you please explain the small green area under the curve again? So this was the significant value.
Yes, of course. Just one moment to regenerate it. Okay. Let's check this. Okay. This is So this marked area in here, it states the difference. So if the difference is statistically significant, the difference in between the target average and non-target average. The target average means the trials when the user looked at a flashing character, for instance, and the non-target ones is when the other characters flashed when to which the user was not gazing. So we are doing a statistical analysis, and the blue part, it's highly the blue patch means under this curve means that the difference is highly statistically significant. So the p-value it's below 0.01, and the green one in here, for this one, the p-value is below 0.05. And it's the same, so this is a positive difference with the green and blue, and it's negative difference when the green line is below the blue line. So it's negative difference, and light gray, it's p-value below 0.05, and dark gray, it's below 1%, below 0.01.
I think it's clear. Thank you very much.
Okay.
I interrupted you before while you were showing something. Already done. Any other questions for Danut? Otherwise, Danut, thank you very much for the nice demo.
Thank you too.
So it's also nice to see step by step how the EEG data is analyzed. Maybe Danut, you could also show the MATLAB command window in the background.
Of course.
Just want to point it out again, because here is whatever Danut showed with the graphical user interface is reported. And this is super easy to copy and paste it into a MATLAB script, and then you can easily do the analysis later on, and you don't have to repeat all these manual steps again. You can just replace the patient or the subject number and run it again. So this is super easy. So thank you very much, Danut, for the nice lecture.